Okay, so what we need to consider is the root of the question. So in the Krebs cycle, which takes place in the matrix, releases hydrogen ions. Okay, so releasing hydrogen ions, that's an important point. These ions, so we're still talking about hydrogen ions, provide a source of energy for the synthesis of ATP using coenzymes and carrier proteins. So describe the roles of the coenzymes and carrier proteins in the synthesis of ATP. If we just go to have a look at the processes involved. If we think in the Krebs cycle, which goes from 6 carbon to 5 carbon to 4 carbon and then back over to 6 with acetyl CoA being added. If we look at this process, coming out of each of these steps we have certain numbers of NADH and FADH. It's not particularly important for this question where in the steps this occurs, but we can see the addition of the hydrogen ions, and that's the ions that are being referred to in the first part of the question. So we start our answer saying that the hydrogen ions from the Krebs cycle are attached to NAD. Now the NAD and FADH, they both go into the electron transport chain. So what happens in the electron transport chain? Well, if we move across, the ETC, if you remember, has got a series of proteins inside the phospholipid bilayer. Okay, and the NADH is converted to NAD, and the hydrogen ion is released. An electron goes into this and moves along the chain before finally binding with O2. So the hydrogen ion has been removed from NAD. So the NAD has been oxidized. So the NAD is oxidized. Once the NAD has been oxidized, the electrons pass down the electron transport chain. This is where the second part of the answer comes in. The hydrogen ion that's produced moves through the membrane, and this occurs at 3 of the carrier proteins with H+ ions moving. When these H+ ions move back, they do so through another carrier molecule. But this time, each time a H+ ion moves through, this causes the synthesis of ATP. And this happens obviously 3 times for NADH, 2 times for FAD. So if we now consider the mark scheme and have a look at what we've talked about there, we've got NAD and FAD being reduced. We've then got hydrogen ions going from carrier to carrier, electrons being passed on, okay, protons passed into the intermembrane space, they flow back through the enzyme to synthesize ATP from ADP and phosphate.